Hey guys, this is meiosis and the sexual life cycles part 2. We start with a review of some meiosis terms that we're going to be using as we proceed today. This first one is that DNA first, for meiosis to happen, has to replicate, meaning make a copy of itself, condense, and form chromosomes. And those chromosomes are usually in the shape of an X. So here we see this is actually a copy of this one. So here you've got one version and it's replicated and it's still holding together right here at the centromere, right there in the middle. So as we go through meiosis, we're going to be referring to these chromosomes in this shape. There's 2 consecutive cell divisions that take place in meiosis. So we break it into 2 chunks, meiosis 1 and meiosis 2. In meiosis 1, you have a separation of the homologous chromosomes. So these guys right here, meaning one came from mom, one came from dad, will come beside each other and they will separate in meiosis 1. The second thing that happens is the separation of the sister chromatids. So that's where the X will actually divide apart and split apart. And we're going to see that in the diagrams to come. This whole process of meiosis makes 4 unique daughter cells. Those are unique, meaning they're not like each other, and they're also not like the parent cell that they were formed from. So that creates genetic variation even in the gametes that are formed through this process of meiosis, and that will lead to varying offspring. So, for instance, you and a sibling, although you have the same parents, may look different. So when we look at meiosis 1, there are 4 main steps. You might say 5, depending on whether you consider cytokinesis its own step or not. So we've got prophase 1, which is the homologous chromosomes will get next to their pair, meaning the one from mom gets next to the one from dad. And you'll actually get what we call crossing over taking place, where a piece of one will break off and unite with a piece of the other. But right now, they're not pulled apart yet. This is called a tetrad, where you've got technically 4 chromosomes connected to each other because of that crossing over taking place. But you get the nuclear membrane will dissolve, and those DNA strands will condense up so you can see them. Then you've got in metaphase 1, which is the next step, all of those chromosomes will line up beside their homologous pair. So the one from mom is lined up beside the matching one from dad. So they'll all line up in the middle on the equator. Anaphase 1, the homologs will separate from each other. So you'll get the one from mom going to one side and one from dad going the other. But if you notice here in anaphase 1, this chromosome is no longer a complete red one. It's actually got a piece of the blue one on it. So you've got mostly mom's chromosome with a little bit of dad's chromosome attached to it. And that may happen in a varying degree of each one of those homologous pairs. And then in telophase 1 and cytokinesis, we have the pinching in of the cell membrane. The nuclear membranes start to reform, and you get 2 cells. Already you can tell that these are unique to each other. They don't look like each other anymore, and neither one of them looks like the original. In meiosis 2, we start with, and this is going to happen in both of those cells that we just made, but I'm only going to show one of them on here, so you have to think of everything times 2. So we start with the cell that we just made, and the nuclear membrane dissolves. Those chromosomes are still condensed. Notice that these X's have a little bit of the blue one attached to it still. Metaphase 2, they line up in the middle. Anaphase 2, those sister chromatids separate, and they go to opposite ends. And then telophase 2 and cytokinesis, the cell membrane pinches in, nuclear membrane forms. Notice that these 2 do not look like each other, and they obviously don't look like the other 2 as well that are made that I don't show on here. So you end up then with 4 unique cells that look nothing like each other or the original cell that they came from. In meiosis, it leads to this term that I've already used once called genetic variation. There are 3 different types of genetic variation that are expressed or seen in meiosis. The first is independent assortment. This is in metaphase 1, and you can go back and look at metaphase 1. But when those homologous chromosomes are lining up beside each other, they don't have to all line up with the red ones on one side and the blue ones on the other. You could have a red one and then a blue one. And so on this side you would have a blue one and then a red one. So you end up with cells that have some of mom's DNA and some of dad's DNA. And that's independent assortment. They don't have to line up everybody on one side or the other. On crossing over that takes place, which is in prophase 1, where part of those chromosomes from one homolog will connect to the other one. Their legs will literally cross, and then whenever anaphase 1 happens, they will break off and actually change DNA. And that exchange of DNA creates sections of chromosomes that are not like any of the parent chromosomes. And then you have random fertilization. In random fertilization, that's the union of the sperm to the egg. So out of those millions, possibly billions of sperm that are released during that the process of leading to fertilization, only one of those sperm is going to get there. So that's a random chance occurrence, that one of those sperm is going to unite with that egg, so it gives it a very wide range of possibilities, considering that the majority of those sperm are also very unique and not like any of the others. So you get a very wide variety of possibilities that make you who you are. Genetic variation thus contributes to evolution by natural selection, because each offspring has the potential to be very different than its parents or very similar. If the difference is going to help it survive, then it survives and passes its genes down to the next generation. If not, then that difference could be a hindrance and allow it not to survive to the next generation. So genetic variation is key. Genetic variation is always wanted in a biological system. The more genetic variation, the better, and that gives us a better population.